Welcome to join us today to explore the world of animal and plant whole genome sequencing with Novogenes, in which both resequencing and de novo sequencing are implicated. And this is an overview of the webinar content I'll be giving today. Animal and plant whole genome sequencing offers a comprehensive view of the entire genomes. It has been widely applied to both the scientific research in the fields of phylogenetics and population genetics, genome-wide association studies, and agricultural breeding programs. So I will take this timely opportunity to guide you through our optimized entire pipeline of both resequencing and de novo sequencing from the sample submission to bioinformatic analysis. In addition, the Novogene-powered case studies and live Q&A sessions will also be incorporated to help you gain further insights on how this service can be leveraged to address the biological questions of interest. So let's get started. First of all, I will start with the resequencing and Illumina and third-generation sequencing platforms. Resequencing refers to sequencing on the species with available reference genomes, and this could be achieved by Illumina or third-generation sequencing platforms. The sequence rates could be subsequently aligned to the known genetic sequences of reference genomes for variation calling involving SNP, InDel, structural variation, and copy number variations among the diverse individuals and populations. The advanced or customized analysis will be further conducted to address the biological questions of interest. For example, the genetic distortion or breeding to unveil the molecular mechanisms of production, adaptation, disease resistance, and climatic resilience, which could be meaningful in accelerating genetic progress, pathogenesis studies, and species and risk conservation, or to investigate the origin and evolution of species in phylogenetics and population genetic research. Here, we presented our optimized pipeline resequencing from the sample submission to library preparation, sequencing, and bioinformatic analysis. So we conduct the quality control prior to each process of them individually to ensure that the samples or the data are qualified and quantified for following performance. For resequencing, either Illumina or PacBio and nanopore sequencing are optional according to the research purpose for the specific variation callings. To be specific, the Illumina with short length, while highly accurate rates is superior in SNP and InDel detection, and whereas the third-generation sequencing platforms with long read length, in spite of relatively low accuracy, detect the SNP structural variations with high precision by crossing over highly repetitive and complex areas. And this is a detailed information of our sample submission guidelines for DNA samples, which varies according to the sequencing platform and library types. And compared to the Illumina sequencing, a higher sample requirement for third-generation sequencing is requested in terms of sample loading and fragment size. And apart from the DNA samples, we also accept non-hazardous DNA extraction from the disparate tissues. And once the samples are claimed by our lab, we will perform the sample quality control within 24 hours to check the quality and quantity of samples. So at Davis Lab, we offer the manual operations of agarose gel electrophoresis to identify if there's any impurity or degradations, and qubit to determine the concentrations. While at China Lab, the AATF fragment analyzer will be conducted for examination of concentration, purity, and integrities. And for the high molecular weight DNA samples, the pass-field gel electrophoresis will be performed additionally for DNA sense checking. After that, we will proceed the qualified DNA samples into library preparations. According to various options of sequencing platforms, the diverse libraries are constructed. In the next generation sequencing, Illumina universal libraries are efficiently constructed by the segmentation, and this combines the ultrasonic fragmentation that randomly shares the DNA into 350 base pair, and the ligation reactions of Illumina-compatible adapters to the fragment. And after the completion of the adapter ligations, the additional motif, such as the sequencing primer binding set, index, the sequences complementary to the OLIC sequence and flip cells, are introduced through the reduced cycle amplification. And for the third generation sequencing library construction, the random DNA sharing will be applied optionally per the molecular weight of DNA samples. For these high molecular weight DNA samples with fragment size ranging from the 10 kilobase to 20 kilobase, the library construction could be directly performed without DNA sharing to ensure the long read length of subseries. The nanopore library is prepared by attaching a ladder adapter and motor proteins to one strand of DNA. And this motor protein will unzip double-stranded DNA helix into two strands and permit the path of the single strand through the pore. While the smart bell library was produced by ligating the helping adapters into the double-stranded DNA fragments. And after the exonucleate and pure PBBs purification steps, the sequencing primer was annealed to the smart bag templates, followed by the binding of the sequence polymerase to the annealed templates. And after the library construction, the sequencing could be performed optionally on Illumina or third generation sequencing platforms. Here, I summarize their comparison and specifications as guideline for selections. So Illumina allows a paired end sequencing of 150 base pair, in which the read length of it is restrained by the signal decay and defreezing, as well as gradual consumption of reagents. Conversely, the third generation sequencing platforms with relaxed requirement of cycle efficiency enables a long read length over 15 kilobase. And we will highly recommend over 10 times and 20 times sequencing depths for SNP and InDel detection and SV or CMV detections respectively. Nevertheless, the sequencing depth shall be determined by the research purpose of experiment, organism of interest, and the amount of material available. The Illumina sequencing are superior in accurate identification of SNP and InDel. However, it also has drawbacks or amplification bias owing to the deployment of PCR amplifications. Furthermore, limited read length also result in low connectivity and GC bias. On the other hand, the third generation sequencing platform with long read length can span the high repetition and complexity area, providing the good coverage, and as well as detect SVs with high precision without GC or amplification bias. What's more, it offers simultaneous epigenetic characterization during sequencing. Yet, the data throughput of it per cell will be fluctuated in basis of species and sample quantities, and the accuracy of its reads also compromised compared to the next generation sequencing data. And here I presented our standard analysis pipeline for the animal and plant whole genome resequencing. The effective clean data after quality control will be aligned to the reference genomes and enable the variations calling in which the statistics and annotation of variants are offered. And Novogene conduct stringent data quality controls in which the quality evaluation and the data filtering are implicated to ensure the accuracy and reliability of sequencing data are qualified for the downstream bioinformatic analysis. So using the Illumina sequencing as an example, the raw reads are saved in FASTQ format, and it contains the sequencing reads information and the corresponding sequencing quality information. In the quality evaluation, the distribution of sequencing error rate and the quality score and GC contents are presented here to reflect the quality of the sequencing data. And in Illumina sequencing, we promise over 85% Q30 for the past samples, which means over 85% of sequence space with 99.9% correct rate are promised. And this literally exceeded Illumina's official guarantee of 80%. In the data filtering, in order to obtain the qualified clean data, raw reads are trained to filter out the quality reads and contaminated technical sequences, such as adapters, the PCR primer, and fragments thereof. And this figure illustrated 3 criteria that were applied for the data filtering in details. First of all, we will remove the paired reads if either read contains adapter contaminations, or if uncertain nucleotides constitute more than 10% of other read, and also for those paired reads, if the quality nucleotides constitute more than 50% of other read, we are going to determine them as unqualified reads for bioinformatic analysis and remove them as well. Yeah, so it's only the clean reads that are subsequently applied for mapping and variation callings. Then we are going to align the effective sequencing data to the reference sequences. So the Wheeler-Anala software in Illumina sequencing as example. The statistics of mapping rate, sequencing types, and coverages are then analyzed according to the alignment result. And the mapping rate, it reflects the similarity between the samples and reference genomes, while the depth and coverage are the indicator of the evenness and uniformity of sequencing rates. And in addition, the mean depth of each chromosome was illustrated here to further evaluate whether the mapping result is in the quantified normal range for the accurate variation calling. And we provide the band format file of mapping results and corresponding reference genomes and annotation files So the HV browser is highly recommended to visualize these span files. Variations are detected and annotated using the corresponding softwares with filter results by standard criteria to reduce the error rate. And we will provide the statistics results of detection and annotations in a table like this, where the upstream and downstream variants and those in exonic, intronic, and intergenic regions or splicing sites are calculated. And moreover, the results of quality distribution is also presented by assessing the credibility of detect SNPs here as an example. And apart from this, the mutation frequency and distribution of variations in different regions of genomes are also illustrated in our final reports. And it was also to mention that the length distribution of variations are also offered, since the length of the inserts in library construction can impact the structural variation detection significantly. And for the proper visualization of the structural variations and the whole genomes, we present them according to the mutation types with circles presented here. So from the outer to inner is chromosome, SNP, InDel, CMA, and SB. And for SNP or the InDel types, the density and distribution is drawn. While for the SB or CMA type, the location and the size are drawn. So here we show the figure of SNP densities. So after we discussed the InDel pipeline of resequencing thoroughly, I will introduce this Novogene-powered case study. Critical population sequencing provide insight into weight adaptations to you and help you to gain further deep insights and resequencing how the resequencing are leveraged in scientific research to address the specific biological questions of interest. So a brief background. Climate change and growing population are putting the global food supply at risk. The broad-weight triticale species expanded its habitat from a small core area within the Fertile Crescent to the global spectrum of diverse environments within a few millennia. And this remarkable evolutionary success shows a great potential in crop improvement, but it still remains to be elucidated. And the purpose of dissecting its genetic mechanisms of adaptation. In this paper, the scientific researchers from the Chinese Academy of Sciences conducted the whole genome sequencing of triticale species. We summarized the research pipeline of this paper here. So in sampling, they collected 414 weight accessions worldwide with 25 species or subspecies in triticales. Then the extracted DNA samples are proceeded to the 350 base pair PCR-free DNA library for sequencing on the Illumina NovaSeq 6000 and 150 with an overall sequencing coverage of 6 times. And following the alignment of reference genomes and variation callings, they also applied a variety of advanced analysis, including the population structure, linkage disequilibrium, and the selective sweep analysis, etc. Lacking accurate phylogeny of triticals was impeding active investigations with adaptations, and also the rise of it has long been controversial. So in this paper, with hundreds of thousands of randomly chosen SNPs, the population structure analysis has been applied to obtain the robust phylogenesis of the triticals within the A lineage, AB lineage, and D lineage. And this is a phylogenic relationship of critical species presented here. And for the result of it, the authors inferred the individual progenitor of critical species from the phenotypic information. And this accurate phylogeny of this accurate phylogeny of critical and built the evolutionary relationship of it within the most recent 10,000 years, which is pivotal for the genetic decision of its global adaptive success. And to further investigate the evolutionary patterns of global adaptation in bread weight, selective sweep analysis are subsequently conducted, and the performance of it enables the systematic genome screen for the genes involved in the adaptations and allows the researchers to reconstruct the model of diversity recovery in the bread weight. And the shift of the genetic diversity of critical species are illustrated here. And in terms of these results, the authors concluded that its evolutionary success are attributed to its divergent adaptation to the global environment and the convergent adaptations to human breeding selections. And to be exact, the introgression helps the divergent adaptation of the bread weight to the global environment by substantially increasing its genetic diversity. And meanwhile, the evolutionary convergence by human selection also provided a channel of constraints that shapes the adaptation of it. So overall, this paper, by unfolding the genetic necessities of wheat as a global crop, offered other new perspectives and transferring adaptive success across the species for the crop improvement. So after discussion, the resequencing for the species with available reference genomes, now let's shift over to the de novo sequences for this in the absence of annotated genomes. So de novo sequencing is defined as the sequencing of the uncharacterized genomes, and while the hybrid sequencing operated by Illumina and the third-generation sequencing platforms. And it empowers the de novo assembly of sequence reads and then construct the initial whole genomes and the genetic underpinnings to advance the research in all disciplines of animal and plant biologies. So as quoted from the Zhonghuaixiang, a famous Chinese academicians in the genetic breedings, the completion of a species genomes project is a new beginning for this species research and industry. And this is our optimized pipeline of de novo sequencing from the sample submission to the bioinformatic analysis. And this is quite similar to that of resequencing, so I will elaborate it here repeatedly. However, unlike the optional sequencing of Illumina or third-generation sequence platforms, in de novo sequencing, we would highly recommend the hybrid sequencing of Illumina and PacBio or nanopore sequencing to maximize the coverage for high-quality assemblies. So in de novo assemblies, So next generation sequence data will be analyzed for the genome survey to get the sequencing strategies and with high accuracy for the subsequent error pollution. And meanwhile, the PacBio or the nanopore long sequencing rates together with the HASI data will enable the de novo assembly to the contig or the scaffold constructions. To start a de novo assembly project, we will request our clients to fill this inquiry checklist. So clients will have to provide us with the species Latin name, starting materials types. It could be either tissue or cell or DNA samples there. So high molecular weight DNA samples is highly recommended, as well as characteristics features of the genome and capacities. And this may include the genome size, repeat content, diploid or polyploid, heterozygosity, and the GC content. And this information are pivotal for the determination of sequencing strategies. However, in case of the lack of these information, we highly recommend our clients to perform a genome survey and Illumina with over 50 times sequencing depth of 350 base pair insert size libraries. And this genome survey can offer a preliminary global view of the genomes by characterizing genome features and complexities. And in addition to the de novo assemblies, we also offer the genome annotations and comparative genome analysis. Well, if any customized bioinformatic analysis is required, please also specify the requested analysis content in this list. And this is an overview pipeline of the genome service. So after the data quality controls, the chemo analysis will be performed to determine the genome size, which can be used to infer the sequence data and depth, and the heterozygosity rate, which could affect the de novo assembly results. So per our previous experience, the higher heterozygosity rate can compromise the de novo assembly results and the contig constructions. And then the repeat content, that high repeat content can confuse the assembly pathways and making it more challenging for us. And the GC content, the samples with high GC content are not amplified as efficiently, and with this inefficiency is amplified exponentially over several cycles in PCR. It leads to notable inaccuracy and the coverage in Illumina sequences. The preliminary genome assembly will also be performed for the species with genome size less than 3 gigabase. And it can offer the instructive hints for the de novo assemblies, whereas it cannot be used as reference genomes directly. And through the contamination analysis, we can determine if there's any other contaminations in the data of these genomes per the coverage versus the GC's content curve of context. Here, we illustrated the available de novo assembly technologies, including the PacBio, nanopore, and HASI. And the sequence strategies and the depths are actually decided by the de novo assembly request. For the contig construction, the PacBio CLR or ANT with over 80 times sequence depth are highly recommended in the regular assemblies. While the PacBio CCIs with sequencing depth over 30 times is advised for a fan map assembly for the species with over 3 gigabase genome size. And for the scaffold constructions, we typically would suggest over 30 times ONT sequencing and more than 100 times HASI data for both regular and fan map assemblies. And the next generation sequence data of the genome survey could then be used for error polishing and correction in the genome assemblies. And this is a summary and comparison of two third-generation sequencing platforms. So it's PacBio CLR mode, CCS mode, and the nanopores. For the insert size, average read length, data output, and movie collections. And compared to the ONT and CR mode, the high-fidelity de novo assemblies not only has advantage in the turnaround time for the de novo assemblies and high accuracies, but also provide high integrity of the genomes. So the continuity of it is a bit relatively low than the CR mode and ONT. While the recommendation of CLR or ONT is primarily based on the consideration of cost effectiveness and high continuity with the longer read length. And this is an overview of the PacBio and nanopore assemblies pipelines respectively presented here. And both of them are based on the overlapping of a set of various long sequencing rates without gap, and for continuous sequence assembly to construct the context. The error-corrected context could be then categorized and ordered into the chromosomes with orientation assigned by the high-seq technologies and apply the linked rates for elevating scaffold. And in addition to the de novo assemblies, we also offer the genome annotations, including the repeat annotation, structure annotation, function annotation, and non-coding R annotation, and to identify the functional elements along the sequence of the genomes, and thus giving the meaning to it. And it's necessary because the sequencing of DNA produce sequences of unknown functions. And the pipeline of it are presented here. And we request at least 3 RNA samples from the different tissues of same individuals. So after the elaboration of the entire pipeline of de novo sequencing, let's see how it could be accommodated into the scientific research. And here I presented this Novogene-powered case studies, genomic insight into the origin, domestication, and the diversification of Resequencing germ cell. So Resequencing germ cell is regarded as one of the earliest domesticated plants, with mustard mentioned as a condiment from as early as 3000 before Christ. However, the evolutionary history of this ancient allotetraploid species remain uncertain. In this paper, the scientific researchers from the Chinese Academy of Sciences conducted the de novo assembly of it, and resequencing is assembled genomes as reference genomes. So here is a summary of the research pipeline. In the assembling, they extracted the DNA and all samples from the species of interest. And after that, they conducted with 350 base pair standard DNA library, small bell library, high-seq library, and RN libraries for sequencing on Illumina NovaSeq 6000 and 150 and PacBio sequence or hybrid sequencing strategy. And after the de novo assembly and genome annotation, the authors aligned the next generation sequence data to the assembled genomes for the following advanced analysis involving the population structure, GWAS analysis, and selective sweep analysis, etc. And through the application of the PacBio and high-speed genome assembly technologies, they managed to construct a chromosome scan genomes of this elucidated brassical germ cell, in which the 1,897 superscaffolds with an amplitude of 5.87 million base are assembled. And these contexts are categorized and ordered into the 18 chromosome scaffolds. And by using the high-speed data, and they also confirm the pseudo chromosomes and manually adjusted 165 misjoined contexts. The final de novo assembly captures 933.5 million base of genome sequence with 867.3 million base, which is 92.9% of them encoded into the chromosomes. And this de novo assembly of brassical germ cell is actually superior to any previous assembly versions, and in terms of the genome size, contiguities, and anchorage. In addition, the genome annotation is also deployed, and among the 92,878 predicted gene models, so the 95.5% were functionally annotated in the public databases. So the resequencing was then performed, and the next generation sequencing rates are aligned to these assembled genomes to capture the major genetic variations, and for the advanced analysis to reconstruct the evolutionary and domestication history of this diverse agent crop species. By leveraging a variety of advanced analysis of population structure, GWAS analysis, and selective sweep analysis, etc. So also managed to determine its single origins in West Asia about 8,000 to 14,000 years ago, and by natural interspecific hybridization, and invert the putative spread route of it, which is shown here. And this result not only sheds the light on the complex evolutionary and domestication history of the flowering time, seed weight, but also on human selections for the pharmacological diversification in these versatile species. And this paper provided a comprehensive insight into the origin and domestications and a foundation for the genomics-based breeding of brassical germ cell, and also paved the way for the future research and breeding of these morphologically diverse condiments, the oil cell, the leaf stem, and the root vegetable species. At the end, I would like to repeat the resequencing versus de novo sequencing, and both of them are focused on the entire genome and offers comprehensive global view of it. And the resequencing can optionally performed as the Illumina or third generation sequencing platforms, whereas integrating the long read length and short, well, highly accurate read sequencing is highly recommended for the de novo sequencing. The main purpose of resequencing is for the variation callings. However, in the absence of reference genomes, the de novo assembly constructs the reference genomes as genetic underpinnings for the biological research of this species. The resequencing types for the sequencing depth for the resequencing is predominantly determined by the research purpose and the variation callings, ranging from the 10 times to 20 times or higher, while at least 50 times sequence depth is required for de novo sequencing. that it could be up to 100 times per the genome complexity of species and the assembly request. And a variety of advanced analysis are also provided for both of them. If the customized analysis is required, so please specify it on our inquiry checklist, and we can further evaluate this with our backend bioinformatic expertise. So prior to the live Q&A session, I would like to introduce our company, Novogene, to you and elaborate the reasons why choose us from the top commercial competitors. The Novogene, founded in 2011, is a leading global provider of genomic sequencing services and comprehensive solutions. We are dedicated to improve the life science research and human health care by leveraging the cutting-edge genomic technologies, bioinformatic expertise, and high-performance computing clusters. And as the global provider of sequencing services, the Novogene headquartered in Beijing has been growing rapidly over the past few years, with branches in Hong Kong, US, UK, Singapore, Netherlands, and Japan. And we can be your trusted sequencing partner with tens of sequencing experience and corporations covering 99% of the world's top 100 scientific research institutions. With our strong scientific research capabilities, we are conferred with universal praise and high appreciations from our clients, attributed to our capability of offering the superior customer experience. And this slide summarizes how we provide sequencing services with superior customer experience. So we offer the advanced CCI systems to allow our clients to manage and track their project analysis. And we have established our state-of-the-art library in US, where all the next generation sequencing projects are fully processed without the transshipment to China. And our largest sequencing capacity also enable us to run the project of any size with flexible packages for a high quality data exceeding the Illumina official benchmarks, competitive turnaround time, and affordable prices. And we can also incorporate the sequencing results from the array of platforms, build the diverse sequencing strategies and comprehensive solutions, and offer the best sequencing results that are tailored towards each unique project. And apart from this, we are so proud to say that we also launched our superpowered robot scientists for the autonomously intelligent next generation sequencing delivery at our Dallas lab, and to realize a substitution of many operations by the standardized intelligent automation and ensures the highly productive and accurate end-to-end process from the sample quality control to sequencing and bioinformatic analysis by efficiently 24 hours per day, 7 days per week, and 365 days per year. A continuous operation and eliminating the cross-contamination without human interventions. In addition, our world-class data centers that comprise of 180 bioinformatic engineers, overwhelming storage space, and peak computing speed for the support of fast, accurate, and secure data delivery. That's the end of the presentation part. Thanks very much for your attention here. And I would like to move forward to our Q&A sessions. And I'm happy to take the questions. Let's check the live questions that proposed by our audience. All right, so for the first question, I would like to answer this one. because I assume that most of you who will have project with us might be quite interested in as well. So we've got this audience that asked, well, if my samples cannot meet your requirements, especially that you mentioned that how sample requirement is requesting for the third generation sequencing platforms. So per our sample submission guidelines, the past samples will enable us to ensure the unsurpassed data quality with 85% of Q30 or higher and contracted the output amount. However, a hold of a QC results doesn't imply that library or sequencing has to be halted. For example, if the samples are insufficient amounts for the lab preparation, we actually can offer the low input or ultra low input for the library preparation. Alternatively, if the impurity are detected in the DNA samples, the magnetic beads, also column chromatography, are available for the purifications. And our project coordinator will inform the corresponding risk in advance following the QC report and offer the constructive suggestions in terms of the QC results and previous sequence experience for you. So if you have any extraction issues, that you may also consult us for advice. We are always willing to support our clients with our strong scientific expertise. So yes, please do not worry about it. And the next question is going to be this one. This is a bioinformatic information related question, and it's about the mapping rates for the variation quality. This could be vitally important for subsequent advanced analysis. So I would like to answer it and share with you. So this audience asked, while mapping rate would allow the variation qualities, could you please list the potential cause of low mapping rate? We highly recommend a mapping rate ranging from the 70% to 90% for the identification of variants per our experience and previous experience, as long as the sequence risk could be aligned to the reference genomes. It shall enable the variation calling of SNP and InDel. Even under the situations that the sequencing rates are mapped to the reference genomes of another species in the same genomes, into the absence of the annotated genomes. It's just that a higher mapping rate will be able to detect more SNP on the InDel, and implies that more of them could be used for their subsequent advanced analysis, such as population structure or GWAS analysis. So for the potential cause of the mapping rate, it could be related with polyassembled reference genomes or relatively far genetic relationships between the reference and the samples. Alternatively, the treatment to the DNA may also change the sequence, such as the best-fit treatment, and here then affect the mapping rate. Or the samples are contaminated with a species, for example, the contamination of bacteria in plants. Right, so for the next one, I'm going to pick a de novo sequencing related questions. Okay, so I found this one. I understand that this is a predominant issue to be focused on the de novo assembly, so I would like to answer it here. So this audience asked how to verify the reliability of de novo assembly results. So apart from the Contig M50 and scaffold M50 statistics to define the assembly quality per contiguities, we also conduct the various evaluations and de novo assembly results. And this may include the sequencing integrity assessment, the sequencing consistency and accuracy evaluations, as well as the related species collinearity assessment. And owing to the time limitation of the webinar today, I'm going to choose the last question for this session. But if you have any needs of animal and plants whole genome sequencing, please feel free to reach out to your corresponding region sales representatives. And the last question is going to be this one. So we had an audience asked about this. Does the DNA samples for the genome survey and de novo assembly has to be derived from the same individual, since it's genomic DNA samples required for the next generation sequencing, while for the third generation sequencing, the high molecular weight DNA samples is requested. Right, so theoretically, yes. The DNA samples for genome survey and for the genome survey and de novo sequencing has to be extracted from the same individual. However, if the DNA samples extracted from one individual are not sufficient for the hybrid sequencing of the entire de novo assembly project, we would like to suggest our clients to sequencing the DNA samples that from one individual on the Illumina sequencing, yet use the DNA samples that derive from another individual will belong to the same species for the third generation sequences. And thanks very much for taking your time today. I'm so glad to share our optimized pipeline and sequencing sequencing experience with you.